Good evening, everyone. Welcome to tonight's presentation of Language and Taxonomy, or How to Get a Species Named After You from Planet Word. I'm Rebecca Roberts. I'm the curator of programming here at Planet Word. And this Language and series is made possible from generous support from the Jack Joseph and Morton Mandel Foundation. And the premise of this series is that we explore the intersection of language and kind of anything you can imagine. It's actually one of my favorite parts of my job is imagining all of the different things that language intersects with, because the list is unending. If you ever have an idea of an exploration of language and, I'd love to hear it. You could put it in the chat now if you want to, or if you follow us on social media or get our newsletter, we always want to hear from you. If you found out about this program from some other source, you're not a regular Planet Word audience member, we'd love to have you. We always post the events on the website, but that newsletter and the social media is really the best place to find out about things, planetwordmuseum.org. If you are a regular, especially if you're a member, thank you very much for that support. It is vital for us to be able to continue to keep the museum free, bring programs to you, do field trips, all the things that we do as a museum. It is my absolute delight to introduce you all to science writer Alison Koontz. As I said, I love exploring the intersection of language and other things, and I read a column she wrote called How to Get a Species Named After You. And I thought, that's it, language and taxonomy. Let's explore that. And she's joining us from a classroom at Cal State Northridge. Alison Koontz, thank you so much for being here.
Thank you so much for having me. So yeah, I have a little presentation, so I'm going to share my screen and let's get started. Hi, my name is Alison Koontz. I am a postdoctoral research fellow at Cal State Northridge. I got my PhD about a year ago, and I also work as a lecturer and freelance as a science writer. And so I really love science. I really love communicating science, and I hope that I've put together a little presentation for you that will give you a little bit of an insight into some of the research that I did early in my career as a taxonomist. So this is not a very science heavy talk, but if you have any questions about science or the language of it, please feel free to ask at the end. So today I'm going to be talking about the intersection of language and taxonomy. I will be going a little bit into the history of taxonomy, into an introduction into sort of the past and sort of what was established many years ago and why we even have the concept of taxonomy. I'll then be going a little bit into the field of modern taxonomy, what we do nowadays as taxonomists, a little bit about the process of describing a new species, why we do it, etc. I will then go into probably the most fun part, which is the aspect of choosing the name for a species, which is always fun. And, people always want to have a species named after them. And so I'll try and teach you how to get a species named after you. You might need a little bit of cash, but that's okay. And then to finish, I will be talking about sort of the general big why. So I get asked a lot why we even do taxonomy as a science. It doesn't cure cancer per se. It's not, it's not very sexy or attractive to some people. But I would like to at least sort of address this question and give my opinion of why taxonomy is a valuable and viable science to continue pursuing in the modern age. So let's get started. Okay, so the goal of taxonomy is basically to just describe and categorize the diversity of the natural world, including plants, animals, and microorganisms. So the history of taxonomy basically dates back to the origin of mankind and of human language. There has always been a need to describe to others within a group what the natural environment and what you are seeing around you is like, especially within the context of a hunter-gatherer or early civilization, where you're going to be out sort of more integrated into the wilderness and the environment than we are currently nowadays. And the idea of being able to communicate with other members about the animals, the flora, the fauna, and sort of the general ecosystem around you is going to be really important. This is generally called folk taxonomy, which is kind of a catch-all term for the common names that we use every day. It's called folk taxonomy, in opposition to scientific taxonomy, which is the Latin names that I'll be talking about later. But folk taxonomy has been around, arguably, I think, since the beginning of human language, because it has always been important to communicate with other members of a group. Humans are social creatures, so this is a very important aspect of our survival. So for instance, this is just a classic forest food web. Imagine you're a hunter-gatherer group who is going out to forage or hunt in the forest that you live in. There are going to be numerous different aspects of the ecosystem around you that you will need to communicate with each other. There are going to be obviously the animals that you are hunting, the interactions of those animals, for example, carnivores who are going to be feeding upon rabbits. There are going to be rabbits who are feeding upon the herbaceous plants that you might also want to collect. And all of these are related within the natural environment in a way that could give you hints about where certain food sources will be. So for example, if you see a rabbit chewing on or digging around for something that looks like a root with a spindly white flowers on top, you could assume perhaps they're looking for a carrot. And in that sense, you could know where to dig up the carrots for food for dinner. The same can be said for sort of looking at the interactions of predator-prey and using that as a method to aid in decisions for hunting as well. And so from our earliest periods as a species, we have always needed to communicate about our natural environment with the members of our group in order to aid in our survival. So folk taxonomy has been around for a long time, but obviously we don't have records of everything that back the 200,000 years when the human species evolved. The earliest folk taxonomy evidence that we have comes from about 3000 BC. In the Eastern medicine, there was the emperor named Shen Nung in China who was well known for his research into medicinal plants. He was one of the first people who studied Eastern medicine and made it popular within China, and he also published one of the first encyclopedias for medicinal plants that we still have around today. Another early example is the Ebers Papyrus records, which is the records of medicinal plants that was used by the ancient Egyptians in around 1500 BC. So in particular, a lot of early taxonomy is related to medicinal plants and medicinal purposes and applications to the human life, because obviously that's a really important aspect of the natural properties of the natural system around us. So kind of moving towards a more scientific aspect of taxonomy, we had Aristotle and the ancient Greeks who sort of began formalizing the categorization of the natural world into different groups, into different sort of catch-all terms to sort of take the biodiversity, the large biodiversity that we see every day, and try and categorize it into smaller and smaller groups so that we can sort of parse that large sense of wonder down into more viable concepts. So for example, we have this huge biodiversity in the world, as we see here. Aristotle was one of the first to distinguish between vertebrates and invertebrates. He called them warm-blooded versus no-blooded, or sorry, blooded versus non-blooded, which actually corresponds directly with what we call now as the two clades of vertebrates and invertebrates. He also was the first to assign specific words, specific groups. So for instance, we have here what he calls the viviparous quadrupeds, the oviparous quadrupeds. This is giving live birth rather than having an egg. He categorized birds differently than fishes, and interestingly enough, actually categorized whales as separate from both mammals, or what he the viviparous quadrupeds, and fish, which I think is pretty interesting and also makes sense given the time period. He also was the first to come up with certain words for different groups of animals that we still do use today. So for instance, he was the one that came up with the word crustacean, mollusk, and insects, and these are still commonly used within taxonomy today as kind of a legacy to Aristotle and the early attempt at scientific taxonomy. So these early taxonomic classifications were trying to categorize the large scheme of the world into observable similarities and differences within morphologies or visible structures of the animals and plants that they were looking at. We didn't look at microorganisms yet because there weren't any microscopes. But the advent of the optic lens in the 16th century led to a more complex descriptions of plants and animals because we were able to look more closely into the microscopic details of what's sort of going on beneath the surface. And also we began finding microorganisms, and the taxonomy of microorganisms is not something I know quite a lot about, but that is, you know, a huge field as well. I just won't be talking about it today. So, but the original problem with this scientific taxonomy, which I think is generally kind of a problem with science in general, is that sometimes we tend to make things as scientists more complicated than they need to be. So for example, in the early 18th century, the honeybee, which, you know, honeybee is a very easy thing to say. The scientific name for the honeybee was this very long phrase that I've put here, and I'm not going to say it, but the basic idea of these scientific names were just using Latin descriptors in a sequence to try and describe as detailed as possible the animal in its morphological distinctions, so that you could distinguish it from other animals. The problem, though, is that no one is going to remember this scientific name off the top of their head because it's very long and very cumbersome. And so scientific taxonomy was kind of stalled by this sort of complexity that was pushed upon it by scientists at the time. This did not change until around the 1753, when Carl Linnaeus, who is a Swedish scientist, began publishing works that basically changed the taxonomy world and introduced something called binomial nomenclature for species names. So today, the taxonomy that we use is called the Linnaean taxonomic system. And the general concept of this is, instead of having a whole phrase or basically a sentence of Latin words that is going to describe the distinction of the species, you're going to just have two names. One is going to be a definition of the genus of the species, which is sort of a group of species that is similar to each other. So it's a sort of a zoom out version of a species. So that's going to be the first part of the name, and the second part of the name is going to be specific to the species itself. And they both obviously were going to be Latin. So under this system, the honeybee scientific name is now Apis mellifera, which is a much easier name to remember and has, you know, continued on to this day. So because of the simplicity and because of the rational organization of the taxonomic system behind this, the Linnaean system has progressed and sort of been the touchstone for taxonomy since the 1700s. Linnaean classifications also included larger scales. So I think the biggest thing that came out of it was obviously the scientific names for species, but we also have these larger classifications that, again, sort of serve to take a wide array of diversity and try and parse it down based on similarities until you can get to the smallest, I guess, point, which is the species. So we have these different organizational phrases called domain, the kingdom, phylum, class, order, family, genus, and species. This goes down from broad descriptions. So domains are basically, are you eukaryotic or are you prokaryotic? Some very basic sort of yes or no binary questions. And these go down further and further until you get to specific descriptions, which are at the species level. So for example, if we are going to take the classic dog and we are going to categorize it based on this Linnaean classification system, the name for the dog is going to be Canis familiaris, but behind that, there's also these sort of nested systems of classification that are going to be descriptions describing various aspects of this species to those who sort of know about it. So for example, we know from these different classifications that the dog is eukaryotic. It is an animal and it has it's chordate, so it has a like spinal cord. It's a mammal, it's carnivorous, and it is part of the Canis family. And these all have certain aspects and descriptions that go along with these specific words. And so looking at these different descriptors, you'll able to sort of easily imagine and classify this animal and what it may look like and what it may like this physiology might be based on just these very simple classifications. So this is just an example of plants, and I think this image is just kind of introductory bio image that I've used before, but it's, I think, a really nice example of showing when you're at the kingdom level, which is one of the higher levels, you have this huge distinct diversity of forms and features and various aspects. You have trees here, you have flowering plants, fruits, etc. These are all categorized as plants, obviously, but there's this incredible diversity here. And as you go through the different groupings and you go down through the phylum, class, order, family, etc., you're able to sort of take this diversity and classify it based on shared characteristics that then further and further classify and condense sort of the knowledge of the plant you are looking at until you get to the species, which is the lowest and most specific categorization. In this case, it's a basic moss rose. So I think this image really just shows how you can take this huge diversity and using these terms and using these classifications, bring the scope down to a very small specific level. So now I'm going to be talking a little bit about modern taxonomy and what we do as taxonomists nowadays. So taxonomy, although we've talked a lot about how it has a long history, it was big in the 1700s, etc. Taxonomy is still a very busy field of science today, and the need for good taxonomy is incredibly high. So for the current estimates for the amount of species of animals and plants on the planet is around 8.7 million, give or take 1.3 million. But only 2 million of these species have been officially described in the entirety of the time the human race has been doing scientific taxonomy. And with increased habitat destruction and mass extinction, there is a dire need to categorize and classify these species before they go extinct for a number of different reasons. So taxonomists have a really big goal in front of them, which is to attempt at least to classify and describe as many species of animals and plants as possible before they eventually go extinct. But the big difference nowadays is that we now have the addition of molecular taxonomy as in addition to sort of the classical characteristics of taxonomy, the descriptors of being different morphologies, different physiologies, different behaviors, stuff that you can physically observe. We also have this addition of the molecular aspect of species as a taxonomic trait. And so right now we have this thing called DNA barcoding is one of the big things in taxonomy right now. Basically, you have a specimen, you take a sample, a DNA sample, and you're able to amplify specific genes that are sort of similar across all animals or all plants. So for example, with animals, we used this gene called 28S, ribosomal rDNA. And the idea for that is that the ribosome is a structure in the body that creates all proteins. And so every animal needs to create proteins. And so everyone has a ribosome. And so sequencing the ribosomal DNA and being able to compare the sequences gives you an idea of, again, of taxonomic diversity on a different level than just the naked eye. So this has led to a large discovery of different species that before were previously thought to be the same. So for example, recent discoveries, I think this is from 2021, we've discovered a new species of skunk. This species of skunk does this weird handstand dance in order to show you that it's about to spray you with its stink. And they found through DNA that there was actually 7 different species of this skunk in a region that was previously only thought to have a single species. This same happened with this bat, and this orange bat in the middle. And I think most interestingly was the same occurred with this species of whale. I think a lot of times when we think of taxonomy, we think of people describing small things like, I'm going to talk about tapeworms, butterflies, bugs, etc. And it's just really funny to me that still nowadays, we can still not understand the species identity of something as giant as a whale, which is completely conspicuous. It's not hidden anywhere, but we've been able to identify these different species nowadays using DNA. So when people first know that I worked in taxonomy, they always ask me, how do you actually get around to naming a species? How do you find a new species, and how do you actually define it and get to name it as a new species? The first step is probably knowing a lot about a very specific group of organisms, and I mean an absolute ton to about very specific, very often marginalized organisms. So becoming a successful taxonomist requires extensive training in anatomy, physiology, morphological characteristics, and at this point also molecular characteristics. And you have to focus on a very small subset of animals, because otherwise, there's such diversity that you can only know so much. And so focusing on a very small subset of animals is kind of crucial to becoming a good taxonomist. So for example, an entomologist is one who studies insects alone. Under the guise entomology is a ton of other subcategories. For example, there's scientists who only work on butterflies, or scientists who only work on beetles, and these have their own names as well. The one that I'm most familiar with is a parasitologist, who is a scientist that works on parasites. And specifically, a cestodologist is one that works only on tapeworms. And so I was a cestodologist for a few years. When I was in undergrad at University of Connecticut, I started working in the lab of Dr. Janine Kyra, who worked on describing and categorizing tapeworms found in elasmobranchs. So this is tapeworms that were only found within sharks and rays, which is a very small subset of all the tapeworms in the world, and really just shows you how you really need to narrow your focus down if you're going to be a good taxonomist. While there, I worked on two separate projects. One of them was amending this genus called Coprobothrium, which was in disarray since like the 1907 or something, and describing a new species to sort of support that new genus. I also worked on erecting a new genus of tapeworms, which involved describing 3 new species for this genus as well. And there's a picture of us at the American Society of Parasitologists meeting in New Orleans in 2014. There's a lot of parasitologists in the world, more than you suspect, I think. And so naming a species is difficult, I'm going to say. And I just kind of put this here. This is the description, the basic description of it within the paper that I published of the tapeworm species that I described, Coprobothrium eleonori. And I'm not going to read this. I just put this description here to show you exactly how specific and how much knowledge you have to have about such a small group of organisms in order to actually define a genus or a species properly within the modern scientific context. So this is an image, line drawing I did of the actual tapeworm species itself. That's part of doing taxonomy. You have to have some sort of line drawing. Sometimes they actually let you do photographs, but that's not usually acceptable. But the biggest part is obviously the verbal and sort of the words that you choose in order to describe the species as well. So you want to be able to describe the species in as much detail as possible, so that's anyone who is going to be, for whatever reason, looking later on at tapeworms that are in bamboo sharks from Borneo, you want to know exactly what you should be seeing so that you can either identify or rule out the species, this particular species for the sample that you're looking at. So I'm just going to zoom in and go a little bit more into detail about like the certain words that we used for one region of the tapeworm, probably the most complex region of the tapeworm, which is called the scolex. People often refer to it as the head. It is the most anterior region of the tapeworm. It's called the scolex for a number of different reasons, but there are a number of specific words that have to do with a scolex that are used as descriptors when you are naming a tapeworm species. So for instance, this short description of the scolex has 4 stalked Bothridia. Bothridia are obovoid, pouch-like, with slit-like openings surrounded by anterior and posterior retracted unloculated flaps. So I've highlighted here a couple of words that probably are unfamiliar to the general audience. And these are terms that are specific to tapeworms. Bothridia are specifically these kind of different sections of the scolex. Tapeworms have various different structures in the scolex in order to attach onto the intestine. Some of them have hooks, some of them have suckers, some of them kind of have these cavities where they sort of latch on to an intestinal villi. And a Bothridia is an example of that. So here I've given kind of one of the primary resources available for describing scolices and different aspects of the Bothridia. So loculi are these little sort of, I call them kind of like accessory parts of the scolex. So you can see here what a loculi looks like. My species was not loculated, therefore it's unloculated. It has 4 stalked Bothridia. So this is the 1 Bothridium, this is 2 Bothridium, 4 Bothridium, etc. It has these muscular bundles, which you can see here within the line drawing, and an apical sucker at the tip of the Bothridia. And so I point this out and give this example not to overwhelm people with science or, the complexities of, specific tapeworms, but just to show examples of how much training you need in order to do taxonomy and the minutia of knowledge you have to have about the structures of the animal or plant that you are looking at. So additionally, not only do you have to have this extensive knowledge, but there's a lot of rules you have to follow when you're actually going around and creating a new species or genus. So the naming and publication of new species is governed by the International Code of Zoological Nomenclature, which is called the ICZN Code. And there are 5 priorities given, 5 steps that you have to achieve in order to successfully name a species that is recognized by the ICZN and will, in that case, go forth in perpetuity. The taxon must be given a name based on the Latin alphabet, and it must be binomial for a new species. The name must be unique, so it can't be, nothing else can be named after your proposed name, which I think is quite obvious. The description must be based on at least 1 name-bearing type specimen. So this is a rule that essentially, when you're describing a new species, you obviously have a suite of specimens that you're looking at in order to describe and do the drawings and these sorts of things. This rule means that you have to have 1 sample specimen deposited as a type specimen within a publicly accessible natural history museum, primarily of the area that you found the species, so that anyone who at a later date wants to, look at the species and wants to verify what they have compared to the species description. They don't need the description or the image that you've given them. There's an actual repository of type specimens for the different species that they can also go look at and verify by eye or by, microscope or these sorts of things. So that's one of the other important aspects. Your species description should include statements about appropriate attributes to define the taxa or differentiate from other taxa. We kind of just went over this. And these first 4 requirements must be published in a work that is obtainable in numerous identical copies as a permanent scientific record. Basically, it has to be a peer-reviewed journal article that describes the new species. You can't just post it on a blog and expect other people to follow it as an actual new species, because the ICZN will not recognize that species. So that's sort of the state of modern taxonomy today. And I'm going to go a little bit now into, once you have a species and you've described it, the most fun part, I think, is what people are most interested in is the naming process of it. People always ask me, oh, how can I get a species named after me? So I'm going to go a little bit into that, and you'll probably find that's more difficult than you hoped. So how do you get a species named after you? Well, the most straightforward thing is to find a new species and name it after yourself. You need a degree in biology as well as extensive taxonomic training to do this. So it's a kind of unviable option And in addition to that, the ICZN, although there is no rule against naming a species after yourself, they specifically say it might be a sign of vanity, which I think is kind of hilarious. So people, upstanding taxonomists, don't generally name species after themselves. It's just not really done. I was trying to find an example just to sort of prove myself wrong while I was researching this, and I couldn't find one. So although it's the most straightforward way and to ensure that you get a new species named after you, it is also in a way the most restricted, because you have to be a good biologist as well as kind of vain and willing to deal with the scorn of the biological taxonomy community afterwards. So there's a couple of other ways to go about this. One of the other ways is to become famous. So a lot of species are actually named after famous people. There's a ton of them. I've just given a few here. There is a insect that is named after Beyonce because it has a golden rear, and they said it was bootylicious, which I think was pretty funny. There's, I think, 9 or 10 species at this point named after Barack Obama, including this spider species. I think there's a slime mold named after him, a lot of different things. And there is a frog that is named after the singer Sting, attributed to his interest in environmentalism and sort of conservation. So if you aren't a biologist or you're not good at biology, another option is to become very famous and have your name attributed to a species as well. Probably, I think the most accessible option is befriending your local zoologist. So many zoologists name their new species after friends, family, and colleagues, especially those who've helped them out in life or in research. So for example, the tapeworm species I named in 2016 is called Copperbothrym eleanori, as a tribute to my late mother, Eleanor. So everyone always asks me, oh, was that sort of, you named your tapeworm species after your mom. Was that a dig on your mom? I was like, no, this is the highest honor I could possibly give anyone. Like, getting a species named after you is, like, in my opinion, one of the coolest things ever. So even though I named a tapeworm species after my mom, it is not a dig at my mother. There's some other examples I know from working within taxonomy for a bit of time. So Dr. Janine Kyra has named at this point more than 200 species. She designated her first new species, Calliobothrym evani, after her friend Evan, who dissected her first shark with her in Baja, California, back when she was first starting out as a tapeworm researcher. So she's also named a ton of species after fishers who have helped her catch sharks and rays, colleagues that have worked with her in describing species, some of the secretaries that have worked with her. There's a lot of people who have helped Dr. Kyra out generally eventually get a tapeworm species named after them. So the moral of the story is, if you know someone who's a zoologist, doing them a favor and being nice to them might get you the honor of a species. I should say that the opposite is also true. There is a lot of evidence of enemies of zoologists getting really ugly and horrific species named after them. So if you make an enemy of a zoologist, you might also get a species named after you. It just might not be the most flattering one. And I'm not sure if that's exactly the avenue that you want. So that's another option. The last option is using cold hard cash. So there's a lot of, well, not a lot, but there is kind of a trend nowadays of auctioning off the rights to naming a species in exchange for money that either goes to a university program or to some sort of conservation program. And it's sort of a fun way of raising money for a cause, while also sort of, getting people involved in biodiversity and taxonomy. So for instance, when I was at the University of Connecticut, the Ecology and Evolutionary Biology department had a prize at one of their auctions called a tapeworm to call your very own. The woman who won was a dentist. She bid $3,000 for the right to name the species, and she named it after her sons, which I think is pretty funny. These auctions are kind of worldwide as well. So in 2018, the Rainforest Trust became kind of famous for auctioning off the rights to name 12 different species found in South America, including 4 frogs, a salamander, a mouse, an ant that had very sharp pincers. These rights ended up going up to $650,000. So a lot of money was raised for purchasing land in South America for conservation purposes. So I guess the easiest, if you really want a species, the easiest option might be to cough up some money, but you might have to actually cough up quite a bit in order to have that honor. So that's sort of a little dive into the politics of species naming. And if you want to have a species named after you, you now have some options. But now I sort of just want to briefly conclude the talk with a look at why we even do this in the first place. So I get a lot of questions about what's even the point of taxonomy. Given the massive rates of habitat destruction and rapid rates of species extinction, the job currently might seem incredibly futile. The truth is, and it's kind of sad, at the current rate of extinction and habitat destruction that is going on, particularly due to man-made causes, we will never be able to categorize all species ever. But that doesn't mean we shouldn't try. The natural world holds many wonders that we have yet to discover, and these wonders and these things that we haven't found yet might eventually be actually beneficial to the human race as well. So people ask me, oh, why are you studying tapeworms when you could be curing cancer? Well, there's a lot of different reasons, but one of them is that this biodiversity can actually benefit humanity in a lot of different ways. So new species mean new avenues for medicine or biotechnology that could actually benefit human health and wellness. So when I was back in elementary school, I was often told that one of the reasons for pressing the government to save the rainforest was that the idea that the cure for cancer might actually be found there and hadn't been found yet. When I was 8, I wrote a letter to President George Bush, who was there at the time, who was president at the time, asking him to please, pretty please, save the rainforest because the cure for cancer might be there. Because my mother passed away from cancer when I was young. So I've always sort of been intrigued by this idea that the rainforest or habitats that are incredibly biodiverse might have things in there that have purpose to human health and to human wellness as well. It's a bit hyperbolic at this point to say that there's a cure, like a catch-all cure for cancer out in the rainforest, but even now scientists are discovering uses for compounds found in rainforest plants to treat specific cancers, as well as plants found in other areas of the world as well. So as recently as 2018, scientists found that a compound found in a rainforest vine species caused drastic alterations in morphology and eventual death of pancreatic cancer cells, and pancreatic cancer is one of the most difficult cancers to treat. And so this was extremely interesting and promising, and just kind of goes to show how even the little plants that you're not particularly concerned about can really have huge medical effects. But I kind of want to end this talk by encouraging people to acknowledge taxonomy as a science, not only for its potential benefits to the human race, but also to kind of preserve the sense of natural wonder that we have for us and our place within the larger world. So not everything that we do as scientists has to be economically or commercially valuable in order for it to have intrinsic value. There is value, in my opinion, in just having beautiful, just having the sense of wonder and the sense of connection to the natural world and your place in it. So this is one of my favorite images. It is a tapeworm species that was described by Dr. Kyra. And I show this, obviously it's been colorized, they don't actually have fluorescent colors, but I show this image just because I think it is an example of something that is truly beautiful in a very bizarre and kind of unique way that only nature can really give us. And I am very happy to have been part of a project that was kind of promoting science for the sake of science and science for the sake of just being interested about the natural world around you and trying to describe and classify things based on, just sort of that intrinsic sense of wonder. So that is all I have to say today. I want to thank you very much for listening to me. Thank you to Planet Word for hosting me. It's been wonderful to give this presentation, to prepare for it. And if you would like to contact me in the future, I have a website, and you can also email me at my personal email as well. So thank you so much.
Thank you so much, Alison. So you mentioned that there's something over 8.5 million species, only 2 million of which have been identified and named. Of the missing ones, what are they mainly?
They're mostly invertebrates. So a lot of them are, I think majority of them are probably going to be insects or worms. So for instance, there's a lot of nematode species that haven't been found because people don't normally dig around for the dirt. Obviously some people do, but a lot of these are invertebrates or microscopic organisms that aren't sort of visible to us in our everyday life. A lot of the mammals have already been described. A lot of larger animals have been described already. As I mentioned earlier, we're still finding new species within those large groups, which is still really interesting, but most of the biodiversity we don't know of is going to be these small, large groups of insects, large groups of invertebrates, these sorts of things.
Yeah. And I love the notion that not only are some species names after someone famous, but there is a reason for it. It isn't just fandom, right? Like the golden butt beetle.
Yeah, that one's my favorite.
Yeah. I saw one that there's a spider named after Johnny Cash, in part because it's black, but also because it's found near Folsom Prison.
Oh, really?
That just struck me as such a clever way to get a name in there that also had some relevance.
Taxonomy has a sense of humor if you look at it closely.
Well, that's the other thing I wanted to ask you about, the sort of revenge naming. That people name ugly species after people they don't like. Is that really a thing?
So Linnaeus was actually really, really famous for doing this. So he was, he described so many different species. He's kind of the father of taxonomy. And so he had, I think, 2 students who he taught and then kind of parted ways with on bad terms. And he named, like, I think a slime mold after, like, one of the species of slime mold has this guy's name, and one of like the ugliest spiders you could possibly find has this other student's name. So he was really big on the revenge naming. I don't know if it's so popular now, but there are some examples out there where you're like, you kind of question whether naming this specific species is an honor or if it's a little bit of a dig. And you can never prove it. You can never prove it either.
It's a very subtle burn.
Yeah.
We've got a couple questions starting out. Someone asks, who maintains the master list of binomials and decides whether a new species binomial is correct or legal? Follow up, what if a taxonomist requests a change to a currently accepted binomial due to new DNA discoveries showing misclassification?
Okay, so for the first part of the question, I think that it is actually, and I could be wrong, but as far as my understanding goes, the list of viable names within a particular group of species is going to be maintained by the scientists themselves. The scientists are going to keep themselves accountable. The scientists are going to know the species names of everything. And so it's kind of this, it's a little bit of a inner circle kind of thing. Obviously, because the papers are out there, there is evidence, and generally for each, like for tapeworms, for instance, there is a encyclopedia base where you can find all these things. But I think for the most part, it's kept within sort of the community. That being said, there is kind of a push right now for a large encyclopedia of life. So there's an encyclopedia of life webpage where people, scientists will put up different species that they know of or have described themselves. And it's trying to sort of accumulate this large encyclopedia of species names. But again, it's kind of a scientist-driven movement. To lead to the second question, which was, I believe, how we do change a species name.
Mm-hmm.
So that is actually part of what my project was, doing that. And the difficult and kind of annoying answer is that you really have to describe the entirety of the species again. And you have to go through the entire process. You have to explain within your redescription exactly why you are redescribing it, why you think that the species name is wrong, why you think it's a different species, et cetera. You have to sort of go through and prove yourself right by creating another publication. And so it's a little difficult, and it's kind of messed up a lot of the times by, no offense, but there's a lot of, especially in entomology, there's a lot of casual scientists who are kind of like rich people who just sort of have big collections and they think that they can name species, and oftentimes they'll publish something but it's wrong. And so people in entomology have this huge problem with kind of having to go through and re-describe things that shouldn't be re-described.
And what about if you wanted to change a name not because of a change in the species but because the name itself has been reconsidered? I'm thinking about, taking down Confederate monuments. I'm sure there are species named after people who were not fantastic people. Is there a rethink of those?
Obviously, that's been something that's come up in recent years. There are a lot of thought pieces about it currently floating around the internet. As far as I know, there is no clear-cut answer to that, besides obviously having a genetic or a morphological basis for renaming the species. But it is something currently being considered because, yeah, obviously there is some problematic names out there. I don't think there's a solution yet, but.
Yeah, well, it also feels with such a backlog of new species that renaming the ones that are already identified.
Yeah, and it could also be a dig, who knows?
Right. Right. We have a question that says, in pre-Linnaean taxonomy, was there a limit to the length of the name? Were names often changed when new distinguishing features were discovered?
That is a great question. I'm not gonna lie, I do not know a lot about pre-Linnaean taxonomy. And so I assume that there's probably, at that point, taxonomy or the science was kind of run by a small group of well-to-do men who kind of did, we're all in connection with each other. And so I think that it was sort of an in-group kind of thing at that point. That's my speculation, but it's not something that I can really comment on.
This notion of, does language have inherent meaning or is it arbitrary, comes up a lot in Planet Word programming. Are there certain sounds that all humans associate with certain things? Or does every language invent its own word for things and there's no sort of commonality of human reaction to those sounds? When you are trying to put a name to something everybody can see, but everybody is seeing it from their own background, their own original language, their own scientific training, do you find that there sort of are universal feelings about how a species should be identified, or is this kind of a free-for-all?
That's a really interesting question. So I think I have two thoughts on that. One of them being that obviously for good and bad reasons, taxonomy is kind of restricted to a specifically trained group of people, and those trained group of people have their own common knowledge, their own common verbiage, etc. And you have to know that in order to be considered capable of describing the new, a species or a genus or what have you. So in that sense, I think that it's a little bit like you have to be, part of it. In the larger sense, I also think that for a lot of these species, names are based on, they're Latin, but their names are based on visible things that you can see. So, different Latin terms come up a lot based on like whether, the color of the species or, the patterning or what have you. So in that sense, there is sort of a commonality and origin for the words to describe what you are seeing. If that answers your question.
Did you have to study Latin?
No, I never studied Latin. That was actually the really interesting part, was I had to kind of learn on the fly different Latin origins. But
yeah, you probably know more than most people at this point.
I'm not sure about that. I know a lot about tapeworms. That's about
it. Someone asks, who has the most species named after him or her?
God, probably Linnaeus.
Linnaeus himself?
Probably, yeah. I think the most common known one is that I know off the top of my head for the modern age is Barack Obama. Like people love naming stuff after Barack Obama, but I think probably historically Linnaeus. Either he named him after himself or other people have named it after him because he's so famous.
And somebody asks, how big are the tapeworms you study? Are you sequencing their DNA?
Yeah, so the tapeworms of sharks, they range quite a lot in size, but the ones that I was working on were really small. So I think maybe one micrometer in length. So they were really tiny. They were really cute, in my opinion. They're beautiful, but yeah, they were very, very small. And you did do DNA sequencing. Nowadays, when you describe new species, and especially new genera, you typically have to back it up with DNA sequencing as well.
I was interested in that detail that a line drawing is preferable to photography. Mm-hmm. What is that about?
I think part of that is kind of like a classic thing. And also when you do line drawings, for instance, you can sort of do like a 3D view. So you can do sort of like a transparency, so you can see the internal like organization as well. And so it's kind of just easier that way. Additionally, though, the photography has come into question recently for a lot of endangered, like new species that are potentially endangered, because when you get a species, you usually have to have a dead sample in order to describe it and to go through it and do the line drawings, etc. But for potentially endangered species, you don't want to do that. And so it's kind of becoming a new thing to allow photography to be an acceptable venue for new species, to kind of protect them.
Yeah. Well, I imagine that would extend to the having to have a specimen available.
Yeah, it's definitely a new, like a hot topic in the taxonomy world.
Yeah, this notion of the process that has to go through from any sort of notion that there might be a difference among species that have been grouped together or identification of a new species. How long does it generally take between a scientist saying, I think this is new and unique, to an actual named species?
I think it depends on how long you've been in the field.
Yeah.
Dr. Cairo could probably do it within like 2 months or something, or even faster. But I think for the general, like for people who are sort of, I guess, laymen in the taxonomy world, it took me 2 years in order to get a publication out. And I think that was pretty quick, actually. So I think it ranges on your expertise and your the amount of time you've spent in the field, but I think a couple of years is kind of the average.
Yeah,
it's the average for most science projects.
In another program, I would love to explore sort of science words that have made their way into lay usage. Mm-hmm. Even genus, species, and general and specific. I want to know which came first, right?
Yeah.
Obviously, they have the same etymology, but do we call things general and specific because of genus and species, or genus and species that way because of those words?
I've wondered that too as well. Yeah.
Well, Alison Koontz, thank you so much for being here and teaching us so much, including the new word scolex. I like there. Yes, exactly. I'm not sure I can slip that one into a conversation.
Probably not.
But it's been a pleasure to hear from you, and thank you so much.
Thank you so much for having me. Have a good night.
Thank you, audience, for being here. We hope to see you at another Planet Word program soon, or live in the museum if you get a chance to come visit us down on 13th and K. Thank you so much. Good night.